So, hello everybody, bem-vindos a mais webinar do ICBAS. It is my pleasure to present Dr. Sandra Weiss. Dr. Weiss is a professor in Department of Community Health Systems and Hpa, endowed chair in mental health in the School of Nursing at the University of California, San Francisco, USA. At this university, she serves as director of the Stress and Depression Laboratory, director of the behavioral research training program in symptom science, and co-director of the Depression Center. She is also vice chair of the board of directors for the National Network of Depression Centers. Dr. Weiss' program of research focuses on causes, mechanisms, and effects of depression, stress, and mood-related symptoms, especially among women and very young children. Much of her current research is examining the impact of fetal and postnatal exposure to a mother's depression and stress on child's development of stress regulation, stress-related biology, and mood problems over the first years of life. Dr. Weiss has over 30 years of interdisciplinary research experience as a principal investigator and collaborator on research grants, and has been a member of and chair of multiple scientific panels for the Center for Scientific Review at the US National Institute of Health. Dr. Weiss has a PhD in biological and development psychology from the University of California at Berkeley, and both a master and doctorate in nursing science from University of California at San Francisco, with a specialization in child and family psychiatric mental health nursing. Dr. Weiss has served in innumerable professional organizations and received several awards. And just to name a few, she's the Living Legend. She received the Living Legend Award from International Society of Psychiatric Nursing, the recognition of outstanding leadership from International Women's Leadership Association, and the Margaret Style Award for Vision and Leadership from Sigma Theta Tau. We are so honored to have you with us tonight. So please, you can take the floor. And Dr. Coralia, the program director of the nursing science PhD, is with us. So I don't know if you want to say something, Coralia?
No, just welcome, and sorry for being late, but I had something else just finishing. So welcome here at our webinars. Thank you for accepting our invitation. And as Maria just said, the floor is yours.
Thank you so much. It's just a real pleasure to be here with you. And I'm going to put up my slides and see if we can share them. Hopefully it'll work. Can you see that yet?
Yes, but you should put on presentation
There
mode.
we go. Okay, yes. Thank you.
But yes.
We'll get it organized here. Does that look okay for you?
Yeah, that's fine.
Great.
That's fine. Thank you very much.
Wonderful. Okay. So I just wanted to start by saying that one of the things that has informed everything I do in my research is all of the work that I have done both clinically and in my previous research as a nurse. My focus is on children and their families, but in particular, I've been working with mothers and young children. And everything that I do in my research, I am constantly reminded of why I do it by my clinical work in nursing. And so what I'm going to be sharing with you today has really evolved from those many years of experience and my ongoing work with mothers and now in particular infants. So as you know, the talk's going to focus on intergenerational transmission of depression and mood disorders. The goals are to, first of all, talk a bit about the status of stress and depression among women during pregnancy and the postpartum, to discuss a model with you for how I really like to understand intergenerational transmission of depression and mood disorders, to identify ways in which mothers' depressive symptoms may contribute to children's emotional distress and mood disorders, and lastly, to discuss the research that I've been working on that suggests a mediating role for various biomarkers in transmission of maternal, perinatal, and postpartum depression to early mood and emotion regulation problems in the child. About 10 to 40% of women in the United States, at least, experience depression during pregnancy and the postpartum, depending on the particular population that's being assessed. And in our recent national study of US women in the postpartum, we found that 29% had depressive symptoms. 15% were mild depression, 8% about moderate depression, and then 6% endorsed severe depressive symptoms. So obviously, depression is a very important aspect of women's lives. And I think from everything that I have talked with my colleagues about internationally and what I've read in the literature, this is true across the world. The model that I really very much like and employ in my own research is one called Developmental Origins of Health and Disease. And this has really informed in many ways how I look at a lot of the variables that we're studying. And this model proposes that in utero exposures can trigger fetal adaptations that really make a huge difference in terms of the long-term outcomes for the child. And such fetal programming can optimize in utero function in the short term. And so it may be a very healthy adaptation when it occurs, but it can contribute to disease in later childhood and adulthood. And this is a graphic picture that shows really how this model looks when we're thinking of mental health in women. And when a woman is experiencing depression or anxiety or any other type of emotional distress, it has physiologic impact on her and it causes physiologic changes. And we're studying this too in my own work. We see this very clearly. And those changes in the woman's body, in turn then, when she's pregnant, are affecting the fetus in the womb. And it's the beginning of what this model would consider the programming aspects of already beginning to make changes in that fetus that when ultimately the baby's born will be set in place as they start to develop. Now, multiple types of adversity that women experience, stressful events or symptoms of anxiety or depression, they can activate stress mediators that then program all of the neural systems underlying cognitive and emotional functioning of the child. And we're looking at, in my own research, how these adversities contribute to a child's early problems in managing stress and in regulating emotions and mood. We recently completed a national study with 750 mothers and infants. And in addition to acquiring measures of depression, we coded videos of interactions of these mothers and infants during free play and used this scale that has been developed by the National Institutes of Health. It's a parent-child interaction scale. And in that study, we found that mother's depressive symptoms clearly shaped her interactions with her child. And if you look at this particular graph, you can see that the columns in green are what's called engagement or positive regard that the mother has for the child. And this is a whole set of items that are a part of this factor. And if you look across the different categories of depression, that was really pretty common for all women, regardless of how depressed they were. They all showed a lot of positive regard and good engagement with their infant. But what we saw is that for the women with severe depression, you can see that in the orange columns, that was the more intrusive negative regard and a group of items that contributed to that. And that's where we really saw an increase for moms who were more severely depressed, in contrast to all the other moms, even those who were moderately depressed, it was significantly higher. And so that when you look at the balance between their negative regard and positive regard, it was pretty even in terms of how they related to the child. So clearly their depressive symptoms were having an impact on how they related to the infant. And when we think about this severe depression that women might have, we've hypothesized that a certain threshold for depression may be occurring, where it's like a tipping point for a woman, when the mothers have much more difficulty managing their symptoms during interactions with their infants. And ultimately, then it interferes with the quality of their care. Whereas with moderate depressive symptoms, the moms still seem able to keep their depressed affect and any negative behavior they may be feeling under control when interacting with their infant. And in my own clinical work, I hear moms say to me, I feel so sad, I feel so upset with the world, but I try not to show it when I'm with my baby. I try to really, be more sensitive and responsive to them. And so we really see that happening, but it just becomes extremely difficult when the woman is feeling severely depressed. So in another study that we just completed, where we looked farther along in the child's developmental span, we looked at children at 2 years of age, and a couple of goals in mind. First, we identified the prevalence of depressive and anxiety disorders for these children at 2 years of age. And these were children who were born prematurely. It's a group of children, population, I should say, of children that I have studied and feel very committed to for, probably about 25 years now. And for a whole variety of reasons, not only the children have many challenges of their own as a result of their prematurity, but in addition, their moms and fathers as well, but they're at higher risk for depression because of all of the challenges that they do face. So we were looking at whether at 2 years of age, these children might already be showing evidence of depressive and anxiety disorders. And there is a clinical diagnostic system that has been developed by an organization called 0 to 3. And we used that in this particular study so that it was really based upon good diagnostic criteria. And then secondly, we determined the extent to which poverty, maternal depressive symptoms, and young motherhood increase the likelihood that the child would develop these disorders in early life. And so what we found is the first row there is just showing you the gender of the children. But for anxiety disorders, 15% of these 2-year-olds already met classification for an anxiety disorder, and another 15% for a depressive disorder. But these weren't the same children now. It's just that it happened that they were both 15%. But then there were children, 6% of them, who met criteria for both anxiety and depression. It's really amazing to think that already that early on, because these disorders are not just that they had a few symptoms, but clearly it was clinically significant as a diagnostic disorder. And then when we looked at these variables to see, well, did maternal depressive symptoms in particular have an influence on the disorder? As you can see that I highlighted there, the children whose moms were depressed had a 2.4 greater odds of developing this depressive disorder at age 2. And then similarly, for an anxiety disorder, the children whose moms were depressed, they had a 5.2 greater odds of developing anxiety disorder by age 2. So we can see that the psychosocial involvement, the psychological aspects of being with a mother who is feeling depressed can have a huge impact on the child. Now, the underlying mechanism for this effect could be, and part of it, as I mentioned, could be the experience of living with the mom, but it also could be genetic or some personality origin. It could involve mom's social modeling of difficulties in mood and stress regulation, or be associated with the quality of caregiving, as I was talking about earlier and what we've seen. But results really do indicate a need for mental health assessment of these high-risk children, starting in infancy, and of course, the availability of treatment for the depressed moms. Now, I'm going to shift to focusing on more of the biomarkers that we've been looking at and some of the sort of biological mediating mechanisms, because though certainly the psychological and social milieu of the child is very important, as I was showing in that model earlier, there's really a belief that in utero, some of these things begin to have a huge physiologic and biologic effect on the fetus, which then ultimately influences the infant after birth. So the 4 different mediating mechanisms that we've been looking at are first the hypothalamic pituitary adrenal axis, which is one of our body's primary stress response systems. And cortisol regulation is really the key variable that we're looking at there. And then the other component of our stress response system, the autonomic nervous system, we're looking at it with a focus on heart rate variability. And then thirdly, we're looking at genetic structure. And though we're also doing some changes to see whether certain types of methylation, epigenetic changes are taking place, what I'm going to talk about today is telomere shortening and the impact of that. And then lastly, we're doing work with the microbiome and composition and diversity of the microbiome and how that may also play a mediating role. So the first, looking at the Hpa axis, that is a huge literature with adults that has shown over the last 10, 15 years that dysregulation of this Hpa axis and cortisol hypersecretion that goes along with it are often risk factors for depression. However, some depressed individuals don't really experience that excessive cortisol release, but just the opposite. They have a more reduced or attenuated cortisol secretion. And these two are sort of seen as different phenotypes of depression at some points in time, because people do respond differently, it appears. But the association of maternal depression in pregnancy with infant cortisol, the studies that have been done have shown mixed findings, and it seems to really depend on the infant population being studied. So this is just a reminder for those of you who may not be working with the Hpa axis every day, but our stress response system, you know, when we experience stressors in our environment, the hypothalamus lets our hormones know, and it starts with corticotropin releasing hormone, that some type of potentially adverse situation has taken place. And then all of the different hormones go through this level of secretion, whereby the pituitary gland and the adrenal gland ultimately then releases cortisol. And the beauty of that is that because it's a peripheral hormone, we can measure it to better understand the stress response. And so we are then looking at cortisol regulation with our infants that we're working with and our younger children. And the way we do that is that we have a stressor that we expose them to. And it's a very ethical stressor that's been used, you know, for many years and is well established. It's called the still face paradigm, where mothers are playing with their child normally and talking, and then for 2 minutes, they sort of freeze. They don't show any affect. They don't talk to the child. And as you can imagine, you know, for some children in particular, this can be a major stressor. But certainly then it stops and they go back to their normal interaction. But what we've been able to do in using this paradigm is to take salivary cortisol samples before this occurs and then after during the recovery period. And that way we can see what cortisol changes occur as a result of being exposed to that stressor. So our second, and I'll get to some of our findings in a little bit, but I'm just going to walk through some of these different variables we're working with. Our second then is infant heart rate variability. And here, our hypothesis is that maternal depression may contribute to dysregulation of the child's developing autonomic nervous system, impairing their ability to adapt to environmental stressors and challenges. And typically, you know, we need to be able to be resilient when we are exposed to things that might make us too aroused. And for little ones who are developing their own stress regulation capacity, this is particularly important. And heart rate variability is really a core measure of ANS regulation, and that's why we've selected to use it. Generally, less heart rate variability is viewed as not a good thing. It's viewed as having less ability to adapt flexibly to the environment as it changes, especially to stressors. And this just sort of shows when we're looking at heart rate variability, the key thing is to be able to see, that the beat to beat intervals there all differ. And that's considered, positive in the fact that they are just responding to changes rather than not being able to dynamically respond when a stressor occurs. And this system, the heart rate variability, is really controlled primarily by the vagus nerve, which is a cranial nerve that controls parasympathetic innervation. And so in the process of looking at the heart rate data, and by the way, in that stressor situation I described before, we also have our little children hooked up to very small little leads in the cardiac monitor so that we're able to acquire their heart rate changes on an ongoing basis. And then we later in the lab are able to go through that in detail and to identify exactly what's happening in terms of any changes that do occur. And we use spectral analysis to be able to look at different rhythms that we might see. And the one that's the biggest concern and of interest to us is this bottom one that says HF. And that's really the high frequency heart rate variability. And it's primarily the measure of the parasympathetic nervous system, or our ability to calm ourselves under stress. And it's one of the more interesting parameters that people look at when they're trying to understand stress response. So then the third biomarker that's of interest here is telomere length. And telomeres are the nucleotide caps that are at the end of chromosomes. And they protect the DNA that's housed within our chromosomes. And it's been proposed that exposure to stressors or other adverse experiences during the intrauterine period can alter or even program the baby's telomere biology. And so that whole time in the womb is seen as a very critical one for telomere development. And newborn telomere length, studies have shown over and over that it appears to be a very key predictor of telomere length across the lifetime. So if the baby starts off with shorter telomeres, not only as adults are they going to have more likelihood of having their telomeres shortened, but it's associated with worse health outcomes. And this is just showing here the telomeres with the little caps at the end, the chromosomes that have the DNA in them, and then they are all within the cell nucleus. And we also acquire these data through salivary samples that we take with the baby, only it's a different type of swab that we use that is for DNA collection. And research has really shown that individuals who have depression may have shorter telomeres compared to non-depressed persons, and that stress causes shortening of those telomeres. But there's very limited research that's looked at this on the effects of prenatal depression on neonatal or infant telomere length. So all of those three variables that I just described, the telomeres, heart rate variability, and then the cortisol, we have looked at those in our structural equation models. And what we found, as you can see, is that maternal depression did predict each of those. However, the relationship between depression and shorter telomeres was much more significant than the effect on heart rate variability. And the relationship between depression and cortisol reactivity, greater cortisol reactivity, was also significant. The strongest relationship of all was between then, because we're looking at how do these factors mediate infant negative mood ultimately. And we base this on coding that we've done. Not only do we code the mother-infant interaction, but we're able to code the infant behavior to get infant mood. And you can see that cortisol reactivity was significantly associated with greater negative mood for the infant and also heart rate variability. Oops. So this really lends support for the whole idea that we're seeing these mediating effects of mother's depression on infant negative mood, and that there are physiologic processes in play that are helping this to happen. As I noted earlier, of course, it's not entirely biologically driven because there's clearly other kinds of things that can make a difference, like the mother's interaction. So then I want to also talk a bit about the microbiome. And this, for those of you who are familiar with the microbiome, it includes the collective genomes of all of the microbes that are living in our bodies. And research has really shown the existence of this bidirectional communication system between the gut microbiome and the brain. And we often don't think about that, how important what's going on in our gut may be to our mental health, and how much our brain has control over what happens in our guts. That we think about a bit more, I believe. And it's all mediated by all these other factors, the neuroendocrine, neuroimmune, and sensory pathways. And there's growing evidence for the importance of this, what's called the gut-brain axis, to mental health, including a link between the characteristics of the gut microbiome and affective problems such as depression. So we have so many species within our microbiomes, and we've been working only with the gut microbiome, but you know, the skin has its microbiome, breast milk has its own microbiome, the mouth has its, you know, on and on. So there are many, many ways that we can look at the genomes of the microbiota in our bodies. And this this slide shows the way in which that gut-brain axis works. And I'm not going to go into details, but as you can see, what's happening in the gut microbiome ultimately heads back over and through things like altering tryptophan metabolism, neuroinflammation, the Hpa axis, which we talked about. All of these things are affected by the microbiome. So there's this constant interplay between the brain, which then also has an impact going back down to the gut. And there's growing research to show support for these very intricate kinds of relationships that exist. So preliminary work suggests that in the womb, a baby's exposure to mom's depression can have implications for early programming of the infant's microbiome, as well as some of those other things. And that future disruption of the baby's ability to adapt to the kind of stressors that occur, and then ultimately have good emotional health, can really be impacted by the microbiome. And our ongoing research has shown that maternal depression explained about 8% of the variance in infant gut microbiota diversity. And when we talk about diversity, it's usually viewed as a very positive thing to have a high level of diversity, because it means that, you know, there are not just limited microbiota, but a balance of the pathogenic and the beneficial, and that there are many, many that will support the functioning of an individual in positive ways. And then also the abundance of species is very important. And when we compared that for moms who were depressed versus not, we found that there were these specific microbiota that were high in moms who were depressed, and others that were very depleted in mothers who were depressed. And this is just showing that, you know, when we compared those depressed moms, you can see down in the right-hand corner that they were very high in these particular species, and yet those weren't present in the moms who were not depressed. And then vice versa, the moms who were not depressed had many bacteroides type microbiota, but they were completely absent in the infants of moms who were depressed. So what do these results suggest? Well, one of the things that we've had to sort of do is to, in trying to understand what the functions of these bacteria might be, is to, you know, look up what they do in the body, how they're related to certain metabolites. And it appears that more severe depression was associated with species that can influence metabolism of steroids in the infant. So corticosteroids are central to the stress response. And so it's having some impact even through the gut microbiome on the infant's ability to metabolize steroids. And distinct differences that we found in the abundance of those species could also influence immune activities and expression of enzymes that are involved in metabolism of steroids. So maternal depression during pregnancy can lead to distinct microbiome changes in early life and influence potentially the trajectory of the microbiome in infancy and beyond. So we're currently collecting data in an NIH-funded study to examine what's called vertical transmission of specific microbial genes from the mom's microbiome to the developing microbiome of the baby during the birth process. And we're very excited to be able to look to see what really is happening in that process and how it might be affecting the child. And so this is just a model that shows what I've been talking about in terms of how we really are trying to understand this process of going from maternal prenatal depression to infant mood disorders. And in this case, looking at the microbial function in the microbiome as a mediating variable. So to sum up then, because I know you wanted to have some time for questions and discussion, our research to date provides preliminary evidence for intergenerational transmission of mood disorders. And we're seeing that maternal depression can induce neuroendocrine, autonomic, and some genetic alterations that appear to be happening in the womb and are affecting the development and integrity of the infant's emotional health. And these kinds of changes can, in turn, create the likelihood of emotional dysregulation and vulnerability to negative mood as the child gets older. So our findings also support the importance of managing a woman's depressive symptoms prenatally to prevent adverse effects on an infant's mood-related biology. And I am a nurse, but this is a very important role for psychiatric mental health nurse practitioners, as well as other mental health professionals. And I don't know what it is like internationally, but certainly in the United States, we see the need, but often there aren't enough resources to refer women to when we do assess the need. So this is really going to be important for us policy-wise to create a better support network for these women. And results also indicate that cortisol, heart rate variability, and telomere length may be useful markers in clinical assessment of risk for mood problems in early childhood. And as I mentioned, for cortisol and telomere length, we do not assess those with blood. They're by using non-invasive samples of saliva, which works very effectively. And for HRV, the heart rate variability, it's a short bout with a cardiac monitor, and gives us a lot of important data about risk. So this is, again, I like all these little models that try to tie all the concepts I'm interested in together. But as you can see, this is really what my program of research is looking at. And I'm fortunate to have some fantastic PhD students and postdocs working in my lab with me who also are excited about having a career looking at these types of concepts and their interrelationships. And so we're really looking to that end point on the right-hand side, find ways that we can then begin to improve emotion regulation for high-risk infants, as well as to prevent mood disorders by being able to intervene in some of these biological processes, as well as intervening at the front end on the left-hand side, where we see that more psychological and social support are needed as well. So that is it in a nutshell, and I'll just take the slides down so we can have questions and discussion. Hello, everyone.
Hello. I don't know what my colleagues arranged for me. Is Margarida taking the lead? Am I taking the lead? I'm not sure. I don't know.
Yes, you are taking the leader.
I am taking the lead. Oh.
Yes, we discussed previously that we will have time for questions and discussion. So, and I think we have at least 2 questions, Corália.
Então anda lá, tu encontraste-as?
Não, posso ficar com elas.
I had one from Afaf.
Yes.
Aha, hello, Dr. Melise.
Dr. Melise is here.
Yes, and she's wearing purple, looks beautiful.
That's it. What else is new, Afaf? Okay, so she asks, is there a regular test for microbiota in pregnant women?
I'm sorry, I didn't hear the first part.
Is there a regular test for microbiota in pregnant women?
Oh, thank you. There is not.
Yeah.
And I think that one of the things that would make that difficult at this point in time is that we don't know which of the microbiota are sort of the critical ones to examine. And so most of the ways in which the microbiome is assessed are these very broad sort of examinations of multiple different genomes in the microbiome. So it isn't to that point yet, but certainly could be.
Yeah. Okay, let me see if I can find the other one. Andrea?
Yes.
Did mode of delivery directly impact the levels found in the neonates? If not, or not studied, would you say that your research points to that and research should look into that? Mode of delivery.
Yes.
And the
So mode of delivery is very important. And we know that even if we just look at the microbiome, if a child is delivered vaginally versus C-section, for instance, their own microbiome looks totally different. And some of the ways in which some of the best beneficial microbiota are passed on to the child is through vaginal delivery. So we are assessing that, we're controlling for that factor, but then assessing the types of microbiota that we see transmitted and what impact they have.
So if I understood right, it's better to have a vaginal delivery?
Yes.
That's what I...
From the perspective of the microbiome.
Yes, yes, for that purpose, of course.
And obviously there are times when that can't happen because of the woman's well-being and whatever.
Yes, no, but I was thinking about, your research and if I understood it right. Yeah, might be. Okay, so Alexandre Correia is asking, if these biomarkers are found in an infant, what can be done in advance to prevent future symptoms?
Oh, that's an excellent question. Well, one of the things that I think is known about telomeres, just taking that as one example, is that even though, as I mentioned, stressors and adverse events can erode those telomeres, those protective caps, research has also shown that they can actually be replenished by certain types of interventions. And there was a landmark study, for instance, that showed that caregivers of very sick people, they had support groups and interventions for them, and their telomeres actually regenerated by a lot of the activities that were a part of this intervention. And they had been in pretty bad shape at the start in the baseline. So I can see that, that same type of thing could certainly happen early on for the infant if that was found. And then, of course, with cortisol, one of the things that we know is that when we work with mothers who are having trouble dealing with the coping of their babies, that we can be successful in helping them learn how to get the baby's arousal down, to regulate their crying in more effective ways, to soothe them in ways they might not have thought about. And all of that can clearly make a difference in terms of the baby's cortisol levels. So I think there are many things that we can do to intervene if we do use any of those as sort of risk assessments.
Ok. Margarida, não encontro mais nenhuma. Aqui mais alguma?
No more questions.
No, but e tu não tens nenhuma?
No.
No, but I have one.
No.
Para lá está aqui outra.
No. We have more questions.
Yes. According to this information, we can assume that a depressive mother have more chances to get a baby with neonatal colics?
That an aggressive mother?
No, a depressive.
Oh, a depressive, I'm sorry.
Depressive.
Okay.
Have more chances to get a baby with neonatal colics.
With colics?
Colics, yes. Tummy ache.
Yeah, I'm sorry, I don't know. I don't understand the colic part.
Elisa Moita. That's the name of.
I'm seeing something in the chat.
Yes, but
Afaf.
intestinal pain. The colics are intestinal pain. That's what somebody said here. You know what colics are for a baby, or there's this expression doesn't exist, or we are having the wrong expression in English.
Well,
when the baby has tummy ache, and he cries like hell.
yes, yes.
Okay, you know that.
Oh, so colic.
Yeah,
I'm
colic.
so sorry. Yes. Do you know, I was thinking you were saying colleagues.
No,
and-
colics. You know,
My fault.
tummy
I'm so
ache.
sorry.
Yes. So the question was, according to this information, we can assume that a depressive mother have more chances to get a baby with neonatal colics.
Yes, I
Yes.
think that may well be true. Absolutely. And I'm sorry it took me so long to get that.
That's perhaps my English as well, but and the sound. Somebody's asking, can we have access to the slides? I think this is going to be Bárbara. Estás aí?
Yes. The slides, the presentation will be available at the end of this first series of webinars. So at 20th of December, we will share with all participants this presentation.
Okay, so that's the a question asked.
We have a new question from Afaf. Yes.
We know that social cultural determinants and environment affect the health and well-being of people globally. Could you speak about what your program of research is doing related to this and some of the results that you find? And in the results that you find, there are differences in intergenerational depression based on ethnicity, cultural backgrounds, work situations,
And so on.
Yes. So, yeah, that is so true. First of all, let me start with the global piece. So I'm not a global researcher. However, I do have colleagues that I work with, and one of them is in Kenya and another is in Ethiopia. And we have done work in both of those areas where we're in particular looking at stress. In Kenya, we have done some very important studies of mothers, actually women who are pregnant, and looked at the stressors that they encountered, as well as their hair cortisol levels, which is a different way of measuring stress response, and found that it had a huge effect on the incidence of preterm birth, the higher the stressors and the higher their hair cortisol levels were. And that's something that we've also found in the United States. And so it's been great to work with my Kenyan colleague to see that that really appears to be a cross-cultural and cross-country kind of finding. And then let's see, the second part of that was, yes. So the impact of like culture and race and sociodemographic characteristics, those are so very important. And we are looking at those in all of our work. But, you know, one of the challenges is that so many of the variables that we're looking at are interacting with one another. And so it's always a matter of controlling, like if we want to see, well, even, you know, if a particular cultural group shows a different finding, we have to control for some of the income level issues and other kinds of adversities in their life to be sure that it's related to culture and not to, you know, some of the other characteristics. But one of the things I would say that is consistent is poverty, and we have found that government assistance are all the women who, in the United States at least, receive government assistance and are below the poverty level. They are much more at risk for depression. And, you know, so there's just such a need there that we really have to meet.
Yeah. Another question? No, there is nothing written on the chat, but I think you already, I just finished examination, if you call it, you know, it's the top of that is not really an examination, but it is. It's the top of our university career for the professors, you know, to be a full professor, a top full professor, you have to go through this thing that I just did in 2 days.
Oh my.
Of a nurse professor, and the specialty is mental health. So, you know, so
great.
I just finished that.
Well, I hope it was a positive review.
Yes, it was very positive. This is extraordinary. And he talked about what nurses can do in all kinds of situations. And now I'm asking you, you know, if you are a nurse in a clinic or there's another question here, not public health, but saúde comunitária, como é que se diz? Community health. What can you do? You know, you cannot do those tests, those chemical tests, perhaps. You don't have the means. So if you feel that the mother might have problems, what can you do? Is there something that a white book or of instructions, you know, to help this mother and child?
Actually, you know, there are a number of different types of interventions that are very effective. And one of them that is used quite substantially throughout the United States is something that's called motivational interviewing. And it's something that it can be learned quite easily. And honestly, it's at the core of nursing, because nurses are educated to do all of these components of, you know, listening very carefully, non-judgmental, compassion, you know, asking the right questions of the person so that they begin to develop an understanding of how they can do something differently. And so these are things that, you know, the only limitation is the time it takes, and that nurses and other health professionals are so, you know, busy in their work that, but other than that, we have the means, and I do believe that there's so much we can do without any kinds of biological assessments. Yes, so.
Okay.
We have 2 more questions.
We have 2 more.
Yes.
Another from Afaf. Interdisciplinary research is vital for the future of science. Could you speak about the challenge and opportunities to develop this kind of team-based research program?
Yes. Well, I want to speak about the opportunities, or the good part first, I guess, because I do work with a whole variety of interdisciplinary colleagues. I mean, for one thing, I work with people in the psychiatry, both psychologists and psychiatrists. I also work with a microbiologist, a molecular microbiologist in my microbiome work. I work with OBGYN folks because it's sort of central to everything we're doing. I work with the neonatologists, and honestly, I have not seen many drawbacks. I have seen just like pluses, because I feel like they learned from me, I learned so much from them, and it's a constant stimulating process. And I think that we end up developing better research, better ideas for clinical work by having these conversations across boundaries of disciplines. And so, I would say the only drawback or limitation to that is that sometimes we have to educate one another about the jargon that we use, or, there've been few cases where I feel that people have assumptions about nurses that they make that are unwarranted, that somehow they don't have the capacities or whatever that the individual thinks they should, but they learn quickly that that's not true. And so it's just a matter of being patient when that kind of thing occurs.
Okay. And there is always hope. At least there was hope for me. I'm a mathematician, so if there is no...
I saw that, actually. I looked up your name and I thought, isn't that wonderful? But you're the dean of nursing though, aren't you?
I'm the dean of the PhD program of nursing.
Oh, okay. Thank you.
And I was the dean of the biomedical school where I worked for 12 years. So I'm used to work with health professionals, but I'm a mathematician. So we can learn, I think, either way. You can learn a bit of statistics and maths, and I can learn a bit of health.
Right.
There is another comment or question. Autism and microbiota, microbiota dysfunction. There was a link in some studies. An autistic child can have more incidence of tummy aches / colics in early ages. My doubt is if a depressive mother has more chances to have an autistic child.
I have not seen any evidence of that. Everything I know about autism is that it is biologically driven. And I do think that the only way in which interactions would come in there would be, that obviously children who are on the autism spectrum, they become stressed easily. And so it is important, for the parents or whoever's working with them to be able to manage their own stress and the stress of the child. But, as far as a cause, if that was what the question was about,
I've not more chances. I don't know.
Yeah.
Yeah. Okay. Elsa Figueiredo says there are also scientific evidence of transgenerational mood disorders in refugee children. You could comment, please?
I wasn't quite sure about whether there's is the question, is there evidence of intergenerational?
There is also scientific evidence of transgenerational mood disorders in refugee children.
In refugee children. Well, that is for sure. The way I think I would view that is that we do know that trauma, when it occurs in one generation, it can be carried over to the next generation, and it occurs in many ways, but trauma and depression are intimately intertwined. And so I do see a lot of that kind of trauma across generations and along with depression.
There are two more questions here, and I think Barbara is making some signs, I think. Barbara, are you making some signs?
No.
Not yet? Okay.
It's okay.
So, Afaf wants to know, on those interdisciplinary teams, who starts? How do you get them started? Who starts?
Well, I love that question because, I would say that in the past, it's been more that nurses would go to others, right? and say, oh gosh, you know, this is an idea I have, and what do you think? But now what's happening, and certainly in the United States, and there's a big push at the National Institutes of Health for this as well. All of research is seen as much more effective if it's transdisciplinary. And so now I see that people are reaching out in so many ways across disciplines to be sure that that's happening. And, you know, I think sometimes anybody who wants to do that, it's a matter of just sort of taking that leap, being a little courageous and sending an email or whatever. I have always had very good responses to that. And I guess now I'm at a point in my career where I'm also getting, you know, people reaching out to me, which is nice, but I think that you have to start someplace. And so just having the courage, I think, to go ahead and contact somebody is what counts.
You can always use Twitter, Afaf. You just tweet and see how it gets.
Yes, yes.
Now it's easier. Now we can send an email. Now we can send, you put it on Facebook, on Twitter, on all those things, you know. 20 years ago, we had to talk to people on the phone, not cell phone, on the phone, or write to them a letter, and it was more complicated. Now I think it's a bit easier, and who starts? Perhaps is a question who thinks about it and says, I think there is a problem here. Who wants to help me think about it? And yeah. I think so. That's my idea as well. João is asking also, João Estevão, do you know whether there is any communication programs for information of healthcare professionals in this field about the last scientific discoveries in this field? If there is any communication program for informations on healthcare professionals?
So,
is there any links that people can look
like
at?
publications or something like that? Any particular aspect of what I talked about today? Because I'd be glad to send, you know, whoever it is, if they have a specific interest, some links or manuscripts that I'm aware of.
Yes, I can send you the address. I'll ask him, and if and we can. That's how it starts.
Right.
Okay, I'll let him know. I'll figure out who he is, and I'll let him know that you'll answer him personally all about them. Oh, thank you. Afaf is thanking me and Maria for having this webinar. Thank you. Thank you, Professor Weiss. And if I'm still thinking straight, Afaf, you sent us her name, didn't you? Didn't you? Yes, I think she did. I think she did. And so it was wonderful. And I think, João, exactly. I mean, that João, how can doctors or professional healthcare takers can take information about the last research published in this field, like mentors or people that are responsible to communicate this information to the professionals to keep them updated? That's what João wants to know. And I'll send him an email with your email.
Okay.
Okay.
Sounds great. Yes.
Serve assim, João. We have messages of wonderful talks, and thank you very much all for being here and all for making this. It was a wonderful talk, great presentation, fantastic talk. I'm reading. Thank you, Dr. Weiss. Thank you. So we hope to see you next time on the next series with some more interesting points of this. And I think this is Alexandra Moreira. She is behind this organization. as well, and I think she can be proud that this school is what we wanted to be, a multidisciplinary school. It was one of the dreams of one of our founders to see a multidisciplinary team. And I think nurses were the later ones to come in because of politics and whatnot, but I think they are now in our school, and I think we are doing a good job, and you are helping us so very much, and we thank you very, very much for everything. Alexandra, are you happy?
Thank you, Coralia. I'm very, very happy. And thank you, Sandra, for the great talk and for coming here online.
Oh, it was my pleasure.
And they are also
the merit is all yours, and the merit is all of the hosts, so Coralia and Margarida, because without you guys, we wouldn't manage to do it. So thank you all.
They are asking if you want any Portuguese collaborator.
Yeah, Portuguese collaborators are very welcome as well, and we want to go forward with both national and international collaborators on this task. I think it's very important to put everybody together on the same boat.
Yeah.
Yes.
For a better world, if we may.
Yeah.
If we may dream.
Yeah.
Okay, yeah. I know it's one of those sentences that perhaps for lots of people don't mean a thing, but I think they do mean a lot. For a better world is what we want.
Yes,
and
I
that's
agree.
what we are trying to do. And I think Professor Nuno Grande would love this if he was around us. So thank you very much. I don't know who's finishing it. Am I finishing it?
Yes.
Thank you, Sandra. Thank you, Afa, for being here. It was wonderful, and everybody that was here. And this is only possible because you are here with us, all of you. Thank you, and we'll have next week another one.
Yeah.
Thank you very much.
Thank you all. Bye-bye.